Well, after studying the promoters, especially the lag promoter, TRP promoter, then the hybrid promoter, next we will discuss about another important element is going to be the ribosomal binding site, in fact. So, because most of the things what we are discussing right now is with the prokaryote, either gram-positive bacteria or a gram-negative bacteria. So, in case of a prokaryotic ribosomes, the ribosomes is made out of the mass. I think based on the Swedberg constant, we can say it is 70s. But this can be dissociated into 50s subunit. Another one is going to be the 30s subunit. 50s subunit has got 23s ribosomal RNA. Another one is 5s ribosomal RNA. Whereas 30s ribosomal subunit has 16s RNA in that system. Alright. So usually what happens is that using a strong promoter, so we are going to transcribe the gene for its mRNA. Once the mRNA is going to be synthesized, then the 30s subunit, ribosomal subunit, will bind to the region for translation purpose, in fact. And again, for the binding, what will be the optimum sequence which is required for the 30s ribosomal subunit to bind, in fact. So now usually what happens is that, now I told you that 30s has got a 16s ribosomal RNA. Now this 60s ribosome, that is the tight binding, that is the 3' region of 16s with the purine rich mRNA is very, very critical. So in this mRNA we should be having good number of purine bases. So now the purine nucleotides. So now the purine nucleotide will bind tightly with 16s ribosomal RNA, especially from the 3' region. That ensures a tight binding, in fact. Like promoters, I told you that there are 160 different promoters, their sequence and its binding with DNA dependent RNA polymerase have been done. In a similar way, Shine and Dalgarno, that is in 1975, so they have also studied the sequences of several mRNA and the binding of this 16s ribosomal RNA with that particular one. So they wanted to understand that, I think, what should be the sequence which is optimum for this one to bind, so that in order to have the increased translation product. So because since she did an extensive work on several ribosomal binding site and its interaction with this 16s ribosomal RNA, so we also call this ribosomal binding site also as an SD sequence, also known as Shine-Dalgarno sequence, because this is very, if you do not have this sequence, even if you do have a transcribed into an mRNA, that mRNA is not going to be translated into the protein. So now this is after the complete analysis, now they have find out certain important rule to become a very good ribosomal binding sequence. Usually the SD sequence is consisting of about like 4 to 9 bases, and suppose if this is the ATG for the first amino acid, so it is positioned between somewhere between 3 and 11 bases from the first AUG. So now this is the upstream, so somewhere between 3 to 11 bases. So now this 4 to 9 bases has been positioned, in fact. Okay, now this is the one. So here we also have to have certain rule. What will be the rule? Rule number 1. The typical primary structure of this ribosomal binding site should have, for example, AGG. Then we have N, can be any nucleotide, 6 to 9 before this first amino acid. Okay, now before this plus 1. So now from here, so now we need to have AGG with several, somewhere between 6 to 9, any nucleotide. So now that is rule number 1. Alright. Rule number 2, suppose if this is plus 1, then we have minus 1, minus 2, minus 3. In the minus 3 position, there is no way we can have G. G should not occur at position minus 3. If there is a G, then the optimal binding is going to be affected. That is rule number 2. Second one is going to be the rule number 3. So now between minus 1 and minus 7, they should contain less than 2 Gs, because we are already in the -3, there is 1 G, we are not supposed to have it. Then between 1 and 7, the maximum number of G, the guanine nucleotide, should be less than 2. So more number of Gs in this region is not good for 30S ribosome for binding. Now most interesting one is going to be the rule number 4, which says at position +5 and at position +10 should occupy either A or T. Normally what we expect that in most of the regulatory elements are always in the upstream region, not in the downstream or in the translational unit. Now even in the translation region, now we don't have a regulatory element, because now at position +5, that means it is in the translation region, and at +10 make sure that we don't have A or T. If there is A or T, then again the protein translation is going to be difficult. So now this is the ribosomal binding site. So what I want to tell you that in case of an expression vector, we need to have the promoter, and promoter should be strictly under our control. So in case of a lac promoter, we have the IPTG. So in case of a TRP promoter, we have the indole 3-propionic acid as an inducer, whereas in case of the PL, it is the temperature. That is very important. Apart from that, we also require a ribosomal binding site. Now the ribosomal binding site, its interaction has been extensively studied with various ribosomal binding sequence by Shine-Dalgarno. So we also call this as an ST sequence, and these are all the important salient features is essential to become a very good ribosomal binding site. So now these are all the things which is required for expression vector. So next we will discuss about the shuttle vectors. Now in case of a shuttle vectors, I told you that we always work with E. coli, especially in the laboratory, because that is easiest to do that one. So in that we have discussed plasmids, Fudge vectors, then we have the cosmid vectors. Then we also discussed about the other drawbacks, especially degradation of aromatic compounds, plant pathogenicity, treatment of toxic waste cannot be done in case of an E. coli. It has to be done by other microorganisms. So now we need to isolate either plasmid from such microorganism, make sure that I think they work together, because we have the bacillus, which is a gram-positive. Basically we discuss about E. coli. Another one is going to be the bacillus subtilis, which is a gram-positive, because they are very important in case of fermentation, actinomycetes, and then the corinoforms in fact. Alright, so now these are the drawbacks which I already discussed. The other drawbacks is going to be that, for example, we have done extensively work with in case of an E. coli, then we take this plasmid, whatever the modification we have done that. Then when we introduce into another suitable host required for the fermentation, one is that host might not take up the plasmid DNA. Even if it takes up, it might not replicate. So even if it takes up and it also undergo replicate, the plasmid bone genes might not express, isn't it? So these are all the different drawbacks from one host to another host. So in order to overcome this, what they have done is that, okay, like how we have identified plasmids in E. coli, so many laboratories also working with other microorganisms which are industrially important, find out whether any of these microorganisms has got its own plasmids. Indeed, they identified several plasmids from different microorganisms. Most of them, the plasmids are going to be very large. Again, that is a disadvantage. Many times they are cryptic, meaning they don't know what is the function of this large plasmid DNA it is there. And many times if it is really large, it is very difficult to handle. So these are all the complications associated with that one. So in order to overcome that, in fact, I think they have successful, and this is one of the natural plasmid. So now this is an RSF 1010 is a natural plasmid. So now this has got an origin of replication. So now this is isolated from the gram-positive bacteria. Okay. So now this one is easily transformed into a gram-positive bacteria, and then it can also undergo replication. So it has totally different origin of replication. Now this particular one also has got an antibiotic resistant gene. So now this is for the streptomycin resistant gene, and this is for the sulfonamide resistant gene. So now these things are a natural plasmid isolated from other than E. coli. I would say that it is going to be the gram-positive bacteria. Now this particular one can replicate in both gram-positive bacteria as well as in case of a gram-negative bacteria. Now this origin of replication. So now what happens, like now you have the E. coli, which is a plasmid, then we have isolated several plasmid from the gram-positive bacteria. Now they are trying to characterize the origin of replication, which can work both in gram-positive bacteria and then gram-negative bacteria. Suppose like this is from a gram-positive bacteria, then these things are going to be resistant. Sometimes in case of a gram-negative bacteria, these genes might not be functional also. So these are the other drawbacks associated with this. But how we can overcome all these drawbacks, so that we have constructed what is known as a shuttle vectors. Now that is the preamble for our shuttle vectors. Alright. So now they have isolated several plasmids from the Staphylococcus aureus, that is a gram-negative, gram-positive bacteria. And whatever the plasmid they have isolated from the Staphylococcus aureus, they were able to transform and expressed in Bacillus subtilis. I told you that Bacillus subtilis is one of the microorganisms extensively used in the fermentation. So that we are working right now the shuttle vectors which works with E. coli and works with Bacillus subtilis. So from the Staphylococcus aureus, they have isolated several plasmids. One is a PUB 1110. So now this particular one offers a kanamycin or a neomycin resistant gene. So now it is 4.5 kilobases. Now PC194 chloramphenicol resistance, PE194 erythromycin resistant, then PT127 tetracycline resistant in fact. So all of them are all Staphylococcus aureus plasmids, but it can be transformed into Bacillus subtilis. Now they want to find out whether we can combine these two things so that it can work both with E. coli as well as with Bacillus subtilis. So now that's what is called as a shuttle vectors. They are also called as a chimeric vectors in fact. Now with this particular background, we will try to understand how various shuttle vectors have been designed. Now the very first vector we can discuss is going to be the vector. Now that is for PHV11. Alright. Now this PHV11 has got a tetracycline resistant gene as well as chloramphenicol resistant gene. So now this can be transformed into Bacillus subtilis in fact. So now here what happens, like now this PHV11 is a combination of PC194, which is a chloramphenicol resistance. Another one is PT127, it is a tetracycline resistant. What they have done is they have taken these two plasmids. So one is PC194, another one is PT127, digested with Hindi3. So now once you digest with Hindi3, so now PC94 will also get a very large fragment with chloramphenicol resistant gene. And PT127 will also give a very large DNA fragments with tetracycline resistant gene. So these two large fragments are combined to give what is called as a PHV11 vector. So now this is a chimera. So now this tetracycline resistant gene is from the PT127 and this chloramphenicol resistant gene is from the PC194. Alright. I will again, sometimes this chlorophene resistant gene is also represented as chloramphenicol acetyl transferase, also called as a CAT gene. I told you in the beginning, because the various antibiotic resistant gene is again by the derivatization of certain protein. In case of ampicillin, it is the lactamase enzyme. Whereas in case of a chloramphenicol, what it produces is going to be an enzyme. That enzyme is called as a chloramphenicol acetyl transferase, also abbreviated as CAT. Now this CAT together with acetyl CoA derivatize chloramphenicol into hydroxyacetoxy chloramphenicol. Now this is no more bind to the ribosomal unit. So some textbook will give us chloramphenicol resistant gene, but in some textbook the same thing is given as a cat. Don't get confused that cat and chloroform resistant gene are totally different. They are the same, but only thing is now here they represent it offer resistance, but some they go ahead and then try to explain the mechanism. So cat as well as chloroform resistant gene is going to be the same. So now we have designed a shuttle vectors consisting of tetracycline resistant gene. Another one is going to be the chloramphenicol resistant gene. So now both can be expressed in Bacillus subtilis as well as in case of the E. coli. So now that is a shuttle vectors. Now the another one is going to be the plasmid PL603. Now this is a combination of both PC194 chloramphenicol, another one is PUB110. Now this is the kanamycin, and now this is the chloramphenicol. So now because usually we know that all these things are all expressed or isolated from the gram-positive bacteria. Sometimes they might not express in E. coli. So in that particular cases, we have to make sure that these ones are all placed next to the E. coli promoters for the expression part of it, in fact. All right. The other shuttle vectors are going to be the genome shuttle vectors. Now that is going to be PHV33. Another one is going to be the PHV23. So first we will discuss about what is PHV33. All right. So now here, so they have taken PC194 Hindi 3, a large segment. So now unlike in the previous one, in fact. Now they have taken this PC194 Hindi 3 large fragment which contains chloramphenicol resistant gene. Now this fragment is inserted into the Hindi 3 site of PBR322. We know that PBR322 has got ampicillin resistant gene, tetracycline resistant gene, then origin of replication. So now here I think we have a PC194. It is again a gram-positive bacteria. It has got an origin of replication. So now this particular one has got the chloramphenicol resistant gene. So now this particular one has got origin of replication to grow individually with E. coli, also origin of replication to grow in Bacillus subtilis. Now this has got tetracycline resistant gene, ampicillin resistant gene and chloramphenicol resistant gene. Whereas in case of an E. coli, chloramphenicol resistant gene, ampicillin resistant gene works fine. But the tetracycline is not working fine. We don't know the reason for that one. Whereas in case of a Bacillus subtilis, chloramphenicol resistant gene is working, ampicillin and tetracycline are sensitive. So in order to make sure that we have to make these also functional, I think we have to use the appropriate promoter from the gram-positive bacteria, that's all. But the construction what they have done in this particular case, so they have made a chimeric one. They can grow both in E. coli as well as in case of a Bacillus subtilis, but they can use an appropriate antibiotic resistant gene for the identification of the clone, in fact. Since they grow both of them in one, we can do initial modification in the E. coli, transfer that into the Bacillus subtilis so that we can take it up for the further fermentation. Now that is the PHV33. Now here we have the PHV23. I think in the previous one we have worked out a shuttle vector, that is PHV11. Okay, now PHV11 we know that is a combination of PC194 plus PT127. So what they have done in case of PHV23 is that, so like PHV33, they have taken the PBR322. To this, they have introduced a large fragment from the PHV11. So now we know that PHV11 is a combination of these two things. So they have taken a bigger fragment from this PHV11 and introduced into the PBR322. So now this is PBR322, again tetracycline, ampicillin. Then again we have tetracycline and chloramphenicol. Now here you see that there are two tetracycline resistant gene. This functions only in gram positive, this functions only in gram negative. So now with this particular one, so again it can be easily grown in both Bacillus subtilis and E. coli, because like here we also have origin of replication from both of them. So now in Bacillus subtilis both these things are resistant, whereas in E. coli, chloramphenicol, tetracycline and ampicillin are offering resistance in fact. So compared to this, now this is going to be a better shuttle vectors. Now you got an idea about like what are shuttle vectors and how those things are designed. And finally, we are going to talk about an expression vector. So here what they have done is that usually now they have taken the kanamycin resistant gene from the PUB110 and they have the CAT. I told you this CAT is nothing but the chloramphenicol resistant gene. So now here what happens is that many times this is not going to be expressed either in E. coli or other things. So what they have done is that they have introduced a promoter from the bacteriophage, SpO2. Alright. Now if you just take the promoter of this particular one, then immediately this can be activated in fact. The most important one is that this particular one is inducible. This will not function all the time. Only when you add 5 microgram per ml of chloramphenicol, then this promoter is going to be activated and this can be induced. I told you in all these shuttle vectors, we don't have a regulatory elements working for that one in fact. So because of that, in case of a shuttle vectors, if you want to have an expression vector, they have identified that an SpO2 promoter is very ideal, and this SpO2 promoter is under the influence of chloramphenicol, so that with 5 microgram per ml of concentration, this is going to be very active. Suppose if you want to introduce any of your gene here, so that it can be, we can do all kind of manipulation in E. coli, but it can be expressed in a Bacillus subtilis, so that we can take it up into further industrial production in fact. So this conclude all the vectors what we are talking with respect to E. coli. Another one is going to be the gram-positive bacteria. Still, the problem is going to be that whether gram-positive or a gram-negative bacteria, they do not have any membrane structures like Golgi or like endoplasmic reticulum or any other things in fact. Suppose if you want to really process a secretory protein, so then E. coli system is not going to work out. You can express a protein, but this requires further in vitro post-translational modification. So as a result, even though we have studied all these activities, still there are certain challenges we have to face. So for that particular challenges, can we find out another cell which has got all these membrane structures? Now that has to be the yeast in fact. We all know that yeast is not a bacteria. Alright. So now all these antibiotic resistant genes do not work in case of a yeast, but yeast has got its own membrane structure in fact. So now developing vector for yeast is another challenging in fact. So I will have some more videos related to the yeast vector in my next videos. Thank you very much.